For this final session of day 2 of the Covering Rare Diseases Fellowship Program, we're going to pivot from talking about gene therapy to the diagnosing and treating rare disorders with precision medicine. And we're joined by Dr. Zeeshan Ahmed, Assistant Professor of Medicine at Rutgers University's Robert Wood Johnson Medical School. So Dr. Zeeshan Ahmed, thank you so much for joining us today.
My pleasure.
So your presentation is going to truly take us into the realm of artificial intelligence. And this whole issue of precision medicine is fascinating to me in that one of our earlier speakers presented it as sort of like making a bespoke suit, or really sort of zeroing in and targeting on what's needed and how to treat it. So please take it away and take us along on your presentation.
I'm grateful for the kind introduction, and I must congratulate National Press Foundation for organizing this wonderful event. And during my talk, we'll be discussing primarily traditional and artificial intelligence-based approaches used for diagnosing and treating rare diseases. So among leading causes of the deaths around the globe and in United States, one can expect cardiovascular diseases, cancer, Alzheimer, diabetes. However, surprisingly, medical error is one of the leading cause of death as well. So, especially when you are looking to study literature related to the life and biomedical sciences, one must, consult authentic literature published through some credible resources. And one of the most credible resources today around the globe is PubMed. And you will be surprised to hear that that over 200,000 articles have been published so far, which reports in different ways medication error. And over approximately 6,000 or 600,000 articles have been published, which includes discussion about delayed treatment. One of the most trending and known subjects in life and medical sciences today is precision medicine. This initiative has been first taken by former President Barack Obama in 2015. And this is what my lab is trying to learn and practice at Rutgers University in New Jersey. We are located in the Institute for Health and affiliated with Robert Wood Johnson Medical School. So the question comes, what is precision medicine and what it makes it special? So talking about precision medicine, it does not only involve the information related to the diagnosis of the patient. However, it is important, but it does include information about patient's lifestyle, eating habits, smoking, exercise, different routine, work routine. And it also includes information about the genomic, phenotypic, and metabolomic profiles of the patient. So in most of the cases, when a patient visits a hospital or clinical environment for any kind of diagnosis, it starts with the symptoms. And if things are not clear, then patient is recommended for different kind of lab tests. And based on the outcome of the lab tests and other requested tests, medications or treatments are provided to the patients. And if the situation is critical, different kind of procedures are involved. So this all process in the clinical setting, is happening, and it generates different kind of healthcare data, which can be used for different kind of analytics to facilitate personalized treatment and predictive diagnostics. However, it is not sufficient enough. So one situation or one question, I can put it here, like one treatment uh, for one patient, for one diagnosis, might not be uh, the same or good or effective for another patient for the symptom, with similar symptoms. So to investigate this cause further, we focus on genomic. We look into the genomic profiles of the patient. We sequence their DNA and RNA. We look at like we investigate those genes which having any kind of mutations might, lead to any kind of disease. Then us taking a step further, we look into the metabolomic profiles of the patients. We study their carbohydrates and lipids and amino acids. We're interested in looking at their glucose level. We want to see the cholesterol, fatty acids, vitamins, and other information, including amino acids. So this information evolves, like right from the genome to epigenome, epigenome to transcriptome, proteome, metabolome, microbiome, epidemiology, pathology, and clinical information, which includes healthcare records in the form of text or written notes, image-based data, and social graphs. So today we are living in an era where we have so much data which have been already produced and available for analysis and precision medicine studies. However, the question might be here that how this data is generated. So starting with investigators, like different kind of investigators in different institutions based on their expertise. They start different kind of studies. They connect with healthcare institutions to get healthcare data or consult or consent patients to get their samples, which could be a serum, blood sample, for example, or tissue-based sample. And then they use those samples in different kind of wet labs or biological labs where they perform different kind of experiments, and they use those outcome and go to different kind of core environments where this could be genomic core, this could be metabolomic core, where they perform different kinds of sequencing and a different kind of data generation. And that data is then maintained or kept within different kinds of computational environments, which nowadays we have a high performance computing environment or cloud computing environment where we have significant amount of processing support, data processing support, and storage capacity. And then it comes to the computational scientists, which are the bioinformaticians, the computer scientists, and different kind of statistical or mathematic experts, which takes this data and perform different kind of analysis. The overall goal is to investigate the disease or diagnosis, perform diagnosis, and support healthcare institution in treating the patients. So when this journey starts, the data generation and analysis and investigation, at each step, the data is changing. The data is no more the same which started from healthcare and coming from core environments, and the outcome, for example, coming from computational side. So one can expect in an ideal world that this is very smooth, data sharing and communication between these different institutions or collaborative institutions is very efficient, and precise treatments or personalized treatments can be provided effectively on time. However, the current situation, even in 2022, is not the same. Every step is very complicated, and the way data has been generated, processed, this overall process even has not been standardized, and it cannot be straightforwardly automated. The data has been exchanged between different units of similar institution in a very complicated way. So this leads us to the rare disease. Like we are in a situation where we are focused on investigating or finding a rare disease. If we focus on the word rare, we can relate to finding something novel or something not discovered completely today. In that situation, just imagine and repeat this whole story in your, you know, head, and you see, like, for example, the patient is coming to hospital, doctors are consulting with the patient, samples have been taken and associated with the relevant study with investigators, different kind of experiments have been performed, different kind of data have been generated from core, maintained within different kind of environment, processed by different bioinformaticians or experts, analyzed, investigated. So just imagine how long this process has taken. And just in case we have a critical disease where patient is in a dangerous situation, we might end up in a situation which is unexpected, or we can lose a life as well. So now then the question comes, okay, guys, we need something automated. So the question is very obvious, how such heterogeneous data can be integrated, analyzed, interpreted, and automated to support precision medicine and translational research in clinical settings. So if we look at what we have today around the globe, especially in the United States, even with all so much effective development around, you know, the story starts with healthcare system, which produce different kind of healthcare data, different kind of research analytics can be involved to support, you know, different kind of treatment. However, different kind of issues within healthcare still remains, like related to misdiagnosis, overtreatment, repetitive treatment based on similar symptoms, decreased productivity, underutilized healthcare data, and significant cost and spending on that. So this is something which is over the water, which is visible for us. What is right now we are doing and expected in future is still under the water, where we are trying to resolve these issues, not these, even more issues than these issues, which I have just mentioned, to provide advanced diagnostics for a personalized patient's treatment, which is economic and which can help us in identifying those biomarkers, which can lead us to the rare and known and critical diseases. So that traditional data analytic might not be very sufficient. And therefore, we are interested in exploring the applications of machine learning to understand the complexities of heterogeneous data, which involves clinical, multi-omics, imaging, demographic, and lifestyle, and other kinds of data sets. So we are focused in fusing these data. We want to integrate this data. We want to study this data alone, as well as in an integrated form. And then the question comes, okay, we want to do that. We want to apply machine learning or artificial intelligence. We want to integrate, we want to investigate. But when we go into the world of artificial intelligence, there are so many approaches available. And if you look at the history, many algorithms from the field of statistics, which are now part of machine learning field, are not originally proposed to support the field of precision medicine. They have been proposed to solve different problems. However, their practice in this domain is also effective in different areas. And which leads us to the question, okay, guys, we have support vector machine, we have deep learning, we have, for example, decision tree, random forest, linear regression, genetic algorithms. So in our situation, which algorithm we should choose? Which methodology we should choose to solve the problem, to help the precision medicine? So this was the question which we started with when we were also interested in applying different machine learning algorithms to study the disease mechanisms, to find the rare disease. So in 2020, we have published an article, so where we have discussed our findings, about availability of most recently published and available machine learning algorithms, which have been applied in the field of healthcare, in the field of life sciences, and utilizing different kinds of data sets, healthcare data sets, genomic data sets, metabolomic data sets, image-based data sets, and applying different machine learning techniques, for example, natural language processing, for example, image processing, for predictive analysis. So I'm glad to share that this study got some good reputation. And this was published by the journal called Database, uh, published by Oxford University Press. And fortunately, over this article is among the high impact research from the journal. Taking a step further, because my lab is not focused on all kinds of data sets. Our primary focus is genomic and clinic and metabolomic data. So we wanted to study, okay, there are many possibilities to study variable kind of data sets, but what are the widely applied possibilities or machine learning algorithms, which are primarily focusing on genomic data and taking a step further, gene expression and variant data for the personalized medicine. So we designed another study, which we have published this year, again with Oxford University Press, one of the journals by Oxford University Press, which is Briefings in Bioinformatics, one of the most reputed journals around. And there we discovered, and reported those algorithms which have been published, within last 5 to 7 years of time span, using gene expression and variant data. And during that study, we were focused on not only identifying the algorithms, but we were focused on like how these algorithms have been applied to gene expression, variant, or different kinds of integrated data sets, and which kind of diseases these algorithms were targeting. For example, cancer disorders, Alzheimer, IBD, cardiovascular disease. So many diseases were there, which we studied during this important project. We have drawn some conclusions as well. Our conclusions include the list of the algorithms, which you can see in this slide. There are many. However, we found the most effective, most popular, and widely adopted algorithms within last few years are support vector machine and random forest. So this led us to a conclusion also, which have been also published in an article recently, that there is not a one single solution from the field of artificial intelligence, which can help us prevention, diagnosis, or treatment using precision medicine. So before I go next, I would like to take a pause here if there are any questions from the audience.
We do have one hand raised. Hawken Miller, go ahead and ask your question, please.
Yeah, thank you so much for your time, Dr. Ahmed. So my story that I'm doing for this fellowship is focusing on data and how that can be used to target therapies for ALS specifically, but we can also go broader with it, and how AI can help understand and come to the causes of ALS, which a lot of it isn't known, and then find treatments for it. So, my question for you is, how could AI be employed, as a way to, find a cause for a condition and go through data and such?
Yeah, I'm grateful for the very interesting question. So before I answer your question, I would like to give you an example. So right now, in the current situation, like for example, I give you my own example of the lab. We are focused on cardiovascular diseases. So for example, going a step further into cardiovascular diseases, we focus on heart failure disease. So there is a set of genes, like maybe 50 to 100, which involves heart failure, right? And having any mutation or high or low or any significant expression in those genes might lead to any kind of cardiovascular disease, especially heart failure disease. But when we draw this study, when we investigate these genes, we do not focus on the rest of the genome of the human. And this is a big question, why? The answer is that so far we have, based on the literature around, we have discovered around over 50,000 protein coding and non-coding genes. So studying using traditional bioinformatic techniques, 50 genes or 100 genes, is not that complicated as if we have to study 50,000 plus genes. So it's not easily and humanly possible. We need some approach which can be used to help this situation. So that's why we are involving machine learning. And coming to your question, that's what we are doing. We are building some training models. And your question also leads to my previous slide, like what is the best solution? There are so many algorithms outside. So which is the best option today to apply to solve your problem? So we have to wisely choose. We have to study the literature. As I have just said, there is not one solution for all the problems. So we have to wisely choose the best option from the machine learning domain, which can help us studying the target disease by utilizing effectively training models. And even then, we might not be able to get the precise solution, but we have to start from somewhere.
Can I ask a follow-up to that real quick?
Please.
Okay, so you mentioned the support vector machine and the random forest models are the most popular. Could you possibly explain that in a bit more detail for those of us who aren't computer scientists?
Yes. So during this study, I have just returned to the slide which you have just referenced to. So in this part, so before I go ahead and address this question, would like me to explain why these are so successful, or what are these algorithms?
Just how it works, essentially.
Okay. So if I go and explain algorithm functional, it might take a lot of time, but I can precisely tell you that these are two completely different methods, and these are mainly used based on the search mechanism to find the appropriate solutions. And the difference between both of them is the major difference, which we have also observed in our lab, is that random forest can be applied to a smaller data set where you have a specific or limited data. However, to efficiently use support vector machine, you might need a bigger data set. So, and their performance is different. So if I have correctly answered.
Okay, that makes sense.
Yeah, thank you.
Shiraz, are you still having problems with your microphone?
I guess now you can hear me.
Yes, I can hear you now. Please ask.
Finally, yeah. Dr. Ahmed, thank you so much. This is Shiraz from Pakistan. I have a very short question considering the available medical possibilities there in my side. Like, do you have any data of the developing countries? I believe that the medical errors probably is one of the major reasons of the casualties over here at the developing countries, as they don't have a mechanism of test. So whatever the test mechanism you just mentioned through the AI system, this is very expensive, and like they don't have even an initial facility. As I'm working right now on Kubeki, which is the first ever case has been reported in Pakistan. So like fortunately the test has been occurred somewhere abroad. Like the Pakistan had no facilities and doctors had no knowledge about this. How do you see this in the form of AI leading it and forwarding it to the data?
Yeah, this is a very important question. So to be very precise, actually the answer is in your question. You have just mentioned that the data is not available. So primarily one of the problems in some areas of the world where medicine is not as much professional as it should be is that there is no healthcare record system available which can be utilized to track the history of the patient. Second, a lot of places in the world, I don't want to name any country or place, but there is no efficient method, or I would say the overall system available, which can support sequencing, because you cannot identify or study rare diseases without, for example, genomic sequencing. And you are also absolutely right. This genomic sequencing is very costly, especially if you are sitting in a place or in a country where you don't have effective or modern sequencing technology. Then you have to definitely send your samples out, and it is a very complicated process. It's not that you can just upload something, to the internet and download from somewhere. It's a pure process. PHI, protected healthcare information, gets involved in that. CLIA is involved in that, and as you're aware that there is no direct solution to that unless there is some improvement in the system level. Yeah, unless there is some improvement in a way of working in hospitals, and also it's also expected that there are some institutions within the country which are doing supporting research. Like there is no country in the world like United States, but there are some countries doing very well. But this problem, I agree with you, is there, and in my opinion, there is no direct solution to that.
Thank you.
Thank you for joining us today, and thank you for this presentation.
Yeah, it was my pleasure, and I am really grateful to you for inviting me today, and I'm so much thankful to all the audience who have participated in this session, and I'm very happy if there will be any questions tomorrow.
And could you please unshare your screen right now in this moment, so that we can get a good view of you as we say goodbye, and we'll see you tomorrow morning.
Thank you so much. Have a wonderful day.